is the cell membrane, it is going to dock with the cell membrane and eventually release all its material inside the cell. Okay, so that's why this method is pretty much acceptable over this particle gun method, because particle gun can damage the cell membrane, the host cell membrane. And also biological vehicles are the most effective ones, such as viruses and bacteria. Now, viruses are genetically engineered so that they cannot replicate inside the host. If they start to replicate inside the host, then they will thrive in huge amount and they will kill the cell, they will kill the host cell and be released. So we need to control their replication. So what we can do, we delete some of the portions from the viral genome which is responsible for the replication of the virus, which is responsible for the virulence factor of the virus. So we cut those virulence genes out and take only the viral packaging proteins within, which helps to package our desired DNA into it, and then we are taking this viral as vehicles to deliver into the host cell. Now, influences on the choice of vector. Now again, I've talked it before that influences will depend definitely on the type of tissue we are targeting, and the second thing is also that the type of response we are trying to get. So we are trying to get a long term response, we use particular type, short term response other types. So we are dealing with high efficiency viral vectors for the gene replacements. If we are dealing with the gene replacement, for example here it is the red color gene, we need to replace it with a green color normal copy. So in this case we must have the high efficiency viral vectors. So we can use this in case of monogenic disorders, because like cystic fibrosis, SCID, hemophilia and something like that. So these are all are the monogenic disorders. Now we can use these high efficiency viral vectors in case of the monogenic disorders, because in those cases simple gene replacement will do the trick. But on the other hand, when you are talking about immune response, in those cases we must have some of the vectors which are having short-term gene expression. We don't need a rapid gene amplification, but still we need short-term gene expression. And also we can use the liposomal delivery system. Okay, because in those cases if it is sensitized to radiotherapy, like sometimes cancer cells, we must rely on the liposomal delivery. In those cases we cannot utilize the viral vectors, because viral vectors are supposed to be very much effective in case of monogenic diseases, but in case of polygenic diseases like cancer, liposomal delivery is much more effective. Now here are some of the viruses and their importance to cause different diseases. Not sorry, not different diseases, to cure different diseases. So you can find adenovirus, adeno-associated virus, and herpes virus, retrovirus, and so on. So depending upon the type of tissue we are targeting, depending upon the target gene sequence we are going to insert into the target cell, we will choose a type of virus. For example, say here adenovirus can hold on to 39 to 38 kb. On the other hand, parvovirus can take only 5 kb. So depending upon the size of our target gene, we can choose it. And also uh, we can see the infection tropism. So dividing non-dividing cells, some of them can only attack dividing cells, something like that, because most of the cells inside our body are where we are doing this therapy are in post mitotic phase, so they usually don't divide like in neural cells, right? So in neural cell therapy we must take those viruses that can infect the non-dividing cells in those cases. Okay. And also packaging capacity is also important depending upon the size of a gene. Suppose our gene of our interest is pretty big, like 28 kb. So in those cases we must take this herpes virus uh, for as our viral vehicle for our delivery. So depending upon these many different things we can take up these different viral vectors, so it's our choice completely. Okay, so viral techniques use various classes of viruses as tool to deliver. We have seen it here in this picture, beautifully illustrated. I won't encourage you to completely memorize this, but you must understand the concept that depending upon the packaging capacity, tissue tropism, and also the expression, transgene expression, whether it is long lasting or short lasting, depending upon our need, we can design this kind of virus. We can take our vehicles. It's simply like that. When you are playing some game, some online game, sometimes you encounter different attacks. Now you need to survive yourselves in those attacks. You have your gun, you have your uh, launcher, you have your mm, a small sword, something like that. So depending upon different times, if your enemy is here very close, you can use sword to kill him, but if it is far away, you must take a gun to kill him. So something like that. And if your enemy is huge, large, you can't kill it with gun, then you use the launcher to kill him. So something like that. So herein again, so it depends upon the type of response we want. It depends upon the type of response, and also it depends upon how we need to cure it, and what is our desire, and what is our desire. Depending upon the type of desire, we'll have limitations, and keeping those limitations in our mind, we are going to choose type of viral delivery vehicle delivery. Okay. Now, the gene therapy vectors are being developed by the genetic modifications of retroviruses, adenoviruses, pox virus, parvovirus, and herpes virus, and other type of viruses we can see here. Okay, so we simply modify their genes. So, what kind of modifications? I have told you before that the modification relies on what we can do. We just cut out the virulent factors out from the cell, from this from the viral genome. Okay, we just keep the part for the production of capsid regions. Okay, so this is where we need to be careful, otherwise the viral can replicate inside the cell. Now the most common viral vectors we utilize are from the retroviral vectors, adenoviral vectors, adeno associated viral vectors, and herpes simplex viral vectors. Now the retroviruses can create double-stranded DNA copies of their RNA genomes. Okay, and can integrate into the genome. So what they are having RNA. So from the RNA they have produced DNA. From the RNA they produce DNA, because they are retrovirus. So they encode the retro sorry, the reverse transcriptase enzyme, and it can transcribe it can produce the DNA from RNA. And for example, we can use it for HIV and Rous sarcoma virus. So HIV, Rous sarcoma virus, and these viruses can be used as the retroviral vectors. On the other hand, adenoviruses are double-stranded DNA virus, so they cause respiratory, intestinal, and eye infections in humans. So see the tissue tropism for them. They are pretty much versatile. They can infect here respiratory tract, intestinal tract, as well as eye regions. So plenty of variety of tissue specific locations can be attacked by adenovirus. So if you use adenovirus as a as a vector for the delivery of our desired gene, we can reach this respiratory tract as well as intestinal tract at one go. So we can take it in those situations. On the other hand, adeno-associated viruses are single-stranded DNA viruses that can insert the genetic material at a specific site in the chromosome 19. Now, if we are having disorders in chromosome 19, any type of disorder, any type of genetic disorder, monogenic, most of the cases, if we are encountering at the chromosome 19, we can use adeno-associated viruses because they are very specific to target the chromosome 19, and they are much more effective to replace those faulty genes at the chromosome number 19. Okay, and herpes simplex virus is also DNA double-stranded DNA virus that can infect neurons. Now this is a very important part, because neurons are most of the time placed in the post-mitotic stage. So the cells are in the post-mitotic phase, they won't divide. So we need to be very careful to give some viral vector which can directly incorporate the gene into the cells, otherwise we can't see the effect. So in those cases we can utilize this herpes simplex virus. So again, it depends on the cell we want to insert our gene. It depends on the size of our target gene to be inserted. It again depends upon the situation we need to deal with to choose a particular type of vector. Now the vector characteristics: unlike the wild type viruses, the vectors are used to transfer therapeutic genes. Okay, by the deleting the essential genes which allow the replication, assembly, or the infections. Okay, now here you can see a concept of packaging cell line, which is very important, because whatever we are having suppose a vector like this virus vector. We need to produce this viral vector. Now how can we produce this type of viral vectors? And for the production of these viral vectors, we must have a packaging cell line where these viral vectors will be packaged, and then we'll take this viral vector to infect the target cell, okay, or the cell which is faulty, or the cell which is expressing the faulty gene, right? So again say so there are some vectors, they lose their ability to reproduce in the target cell and can be replicated in a cell line which supplies the deleted function. Remember this. So here, this is a gene. Suppose we are having a target gene. Now the gene is from the viral system, okay. So suppose we are having a viral gene, so this is the viral gene, and this viral gene is having regions where we can insert our gene of interest. So say this red region is gene of our interest, and other regions are the packaging signal for the viruses. So this psi thing is the packaging signal of our viruses, and this red line is the gene of our interest. Now we need to incorporate this inside a viral vector, right? Now remember, we have talked always that if we are using viral vectors, we must exclude the virulent factors. We must exclude all the necessary factors which will help the virus to replicate and thrive in huge amount inside